Hi friends, welcome again to this tutorial where we will learn in which fields PCR can be used. PCR technique has widespread applications in the field of medicine, research, forensics, agriculture, and many more. Let's learn more about them. But before we begin, let's quickly have a look at the answers of our last tutorial's problems on how to assemble a PCR reaction. So here, for the first question, we know that the number of DNA copies made by 30 PCR cycles will be 2 raised to the power 30. If we begin with one molecule of double-stranded DNA and assume that the PCR reaction ran with 100% efficiency. Hence, for 600 DNA molecules, it will be 7.044 into 10 to the power 11 copies after 30 cycles. For the next part, the PCR reaction worked with 78% efficiency. Hence, we multiply with this factor and the answer will be 5.49 into 10 to the power 11 copies. For the next problem, here the tagged DNA polymerase will be added per reaction. Hence, we will not use the formula C1V1 equals to C2V2. Rather, calculate that if 9 international units are present in 1 microliter, then for 3 international units, we will have to add 1/3 or 0.33 microliter for this reaction. For this problem, we will calculate the volume of primers separately for forward and reverse primers, which will be value V1, as their stock concentration or C1 values are different. Here C2, which is 0.01 micromolar or 10 nanomolar, and V2, which is 10 microliter, are the same for both the primers. Hence, V1 for forward primer will be 1 microliter and V1 for reverse primer will be 0.4 microliter to be added. Now, let's begin with the PCR applications. In our last video, we saw how PCR works based on the biochemical process of replication and how with each cycle of PCR, the amplicons double. This process thus can amplify a tiny amount of DNA into billions of copies. Therefore, PCR, which is rapid, precise, and not very expensive method, is tremendously useful choice for many fields. PCR is used in research like for cloning, site-directed mutagenesis, and sequencing, where we will be focusing today. PCR is also used for genotyping, genetic testing, diagnosis of infectious diseases, forensics, and agricultural biotech, which we will cover in next part. PCR can be used for direct cloning, in which a gene of interest or any other source of DNA whose sequence is known and we wish to clone, it can be amplified using PCR and then inserted into a vector using recombinant DNA technique, as I taught in my 5th video tutorial in this series. We can also add additional nucleotides at 5' end of these primers before inserting them into the vector. These additional nucleotides can be used either to create a new restriction site for cloning or for other purposes. Here in this example, we wish to clone a gene of interest in PET37B vector using XHO1 and KPN1 restriction enzymes, which do not cut the GOI, but also GOI do not have these two restriction sites to help in this cloning. For this purpose, we PCR amplify the gene of interest with a set of primers, where with forward primer we have added a few additional nucleotides to create the recognition sequence of XHO1. And similarly, we have created a KPN1 site at the 5' end of the reverse primer. Now, PCR amplification of the gene of interest will add these extra nucleotides corresponding to new restriction sites, and majority of the PCR product will have them. We can now restriction digest the PCR amplified gene of interest with XHO1 and KPN1 and insert in PET37B MCS for cloning. Yet another application of PCR in cloning will be to select for transformants. Colony PCR can be used to screen the presence of gene of interest in transformed bacteria. In cloning experiments, some plasmid vectors may not receive gene of interest, Hence, they need to be selected. A small portion of transformed bacterial colonies is used, and we can use vector-specific and gene of interest specific primers. To check for the presence or absence of the insert by checking the PCR products on agarose gel electrophoresis, if there is no gene of interest, the PCR product will be of vector size, and if gene of interest has been inserted successfully, we will see a bigger PCR product including the size of the gene of interest. Besides, desired mutations can be introduced into a gene to study its effects on protein structure and function. This process, known as site-directed mutagenesis or SDM, is used in PCR-mediated cloning. We can introduce point mutations within a specific sequence using primers where one base changes to another such that an amino acid changes in the protein product. As you can see here, that forward primer has a purposely changed base which will amplify the recombinant plasmid with desired mutation. These PCR products are then self-ligated to create a new recombinant DNA with point mutation. Or we can introduce deletions within a specific sequence using a set of forward and reverse primers such that their 5' ends are apart by a gap. This gap is the sequence we wish to delete. First, a target sequence is cloned in a vector and then amplified using this primer set. The PCR product thus will have the desired deletion. These PCR products include the target as well as vector sequence and thus can be self-ligated to recircularize and transform the competent cells. Similarly, if we wish to insert a few extra bases in a given target sequence to create an insertion mutant, we can design a primer set with that extra sequence attached to the 3' and 5' ends of the primers in such a way that after PCR amplification, the PCR products have that sequence inserted in both the DNA strands. These PCR products are then self-ligated to recircularize and transform the competent cells. PCR-based SDM is rapid and easy to use. The only crucial step is the appropriate primer design. SANGER'S sequencing method is based on PCR process, where first a set of PCR primers amplify the DNA fragment while attaching a binding site to the 5' ends for the sequencing primer. A sequencing primer then attaches to these tail sequence sites and a SANGER'S sequencing reaction takes place. In next generation sequencing or NGS, a target DNA is first fragmented, then its ends are polished by removing any unpaired bases. Adapters are added to help the primers to bind. Additional nucleotide sequences can also be attached like barcode sequences when multiplexing is performed. In multiplex PCR, several samples can be mixed and run in one single experiment. Illumina's bridge PCR and emulsion PCR of pyrosequencing NGS methods are examples of such sequencing methods. Methylation-specific PCR or MSP can also be used for sequencing of DNA regions where methylation is present. Methylation converts cytosines or C bases into 5-MC, which is 5-methylcytosine, and is commonly found in gene promoter areas. Here we see a sequence of DNA with a methylated region. Two sets of primers are designed to detect methylation. DNA sample is first treated with bisulfite. Bisulfite converts unmethylated cytosines to uracil. If there is any methylation already present at the time of bisulfite treatment, like 5-methylcytosine, then these methylcytosines are protected and not converted to uracil. Then two sets of primers are used based on target sequence. One set has guanines to pair with 5-methylcytosine if present. These are methylation-specific primers. Other set is non-methylation-specific primers and have adenines to pair with uracil. If DNA is methylated, amplification happens from the guanine containing methylated primers. If DNA is unmethylated, the amplification takes place through non-methylation primers. So this was about some of the PCR applications which we saw. The remaining PCR applications I will be discussing in my next video tutorial. If you have any questions and suggestions, please comment. And if you have liked my video, please like and subscribe my channel. My next video on PCR application part 2 will be covering use of PCR in genetic testing, genotyping, diagnosis of infectious diseases like tuberculosis, HIV, and even the SARS-CoV-2 virus, forensics, and in agricultural biotechnology. Don't forget to watch. Thank you and bye-bye.